Have you ever wondered that a banana gives a chocolate flavor? Do you know that insulin and medicines they are actually produced by the different kinds of microbes like fungi and bacteria? Okay, you can come to the agricultural. So all we know that farmers are also producing different kinds of varieties of crops which is disease resistant and drought resistant also. So how it is happened? All it is happened by the help of a rDNA technology, which is very innovative and interesting technique we are going to discussing about. Hi everyone, I am Rajalingam, biology faculty. Okay, today we are going to discussing about rDNA technology. So I can call it as art of Jugaad. Okay, so which is innovative. So shall we go for a topic? So it is actually a very interesting topic. So what are the steps involved in this rDNA technology? So there will be a 5-star rating, 5-star steps. 5 steps only is there. What are they means? Isolation of desired DNA and cutting of desired DNA and plasmid DNA by the help of different kinds of enzymes. They are known as restriction enzymes, and after that joining of desired DNA and plasmid DNA, then we are going to inserting into a suitable bacteria like Escherichia coli. Then after that we need to go for the last step is downstream processing. Okay, so these are the total overall procedure within the rDNA technology. So by this rDNA technology nowadays we are producing lot of medicines and lot of food products also. So here this is actually the overall complete process. So we are taking the bacterial plasmid DNA and whatever the eukaryotic plant or animal chromosome. So here they are cutting by the help of restriction enzymes. Okay, then we are adding we are joining by the help of a ligase and we are introducing into a bacteria. Okay, after that it will be forming lot of colonies. So we know that. Next here some I already explained you the restriction enzymes, they are cutting into a different positions. So some of them are endonucleases, some of them are exonucleases. Endonuclease cuts at specific sites only, which is forming the sticky ends, and exonucleates cutting at the parallel, cutting to form the parallel ends only. These are the different types of restriction enzymes. But first restriction enzyme is Hind2, okay, which is derived actually obtained from the Haemophilus influenza. Okay, so here after the insertion of these bacteria, so we are actually separating whatever the recombinant DNA from the bacteria. Okay, how we are separating means, so these are the transformants and non-transformants. Transformants are containing the rDNA, non-transformants not containing the rDNA. So by the separation of these transformants from non-transformants, we are going for the separation of recombinants from the non-recombinants. So after the recombination separation only we are going for the downstream process. So in this one we are extracting the exact rDNA from the bacteria. This is actually bioreactor. So here huge amount of the rDNA we can extract from the bacteria. Okay, so this is actually bioreactor, and here after the extraction of rDNA we are using to producing by either insulin or medicine products. After that we are going for the clinical trials. After clinical trials only we are sending the marketing. So this is overall process within the rDNA technology. I hope you understand. Thank you very much.